Hi, everyone. Thank you so much for joining us today. We are very excited to have our last Knowledge Portal webinar for 2025. Before we get started, I just want to remind everyone to please feel free to use the chat and Q&A. We will be tracking all of that throughout, so please feel free if you have questions to pause us so we can go over that. So we just have a couple updates as we get started. We are now on Blue Sky. So we have new social media accounts. We know that there is a lot of people in the research space who has been migrating over to Blue Sky. So we have a couple accounts there. So please feel free to join us. It's going to be a lot of the same information, but it's just another avenue for you to get information from our team. And as I had mentioned earlier, this is the last session for 2025. So I wanted to give everyone a preview for what the dates were going to look like for 2026. We are very excited to have another year of incredible presentations and work showcased through this. And so with that, those are my administrative updates. I will hand it over to our team who is going to be presenting on the predicted effector gene aggregation standards and unified schema or Pegasus framework. So I'll hand it over now.
Super. Thanks, Mackenzie. I will get myself set up. I think we want to keep this an active, engaged conversation. So feel free to raise your hand, pop things in the chat, interrupt us. I think you can, well, we have to unmute you. Is that right, Mackenzie?
Yes, so if you
Okay.
raise your hand, I can take you off mute,
Yeah.
and then you can ask your question over audio or use question and answer or the chat.
Right. And then for some of you who are at ASHG, you may have seen some of this content before, but since not all of you could make it to ASHG, we thought we'd do it again. And that's really what today's about. And I'm going to kick us off with a little intro and then hand it off to the real exciting work by Aoife and Laura. So let me just quickly share my screen. And I actually had an original opening plan for this. But then last night I saw this paper and it really spoke to me as to why we should actually open the session with this. So every night I look through various social media feeds and different, alerts I get to emails about new papers that are relevant to what I do as a human geneticist. And this paper popped up last night and it was like mapping the genetic landscape across 14 psychiatric disorders. And I was like, wow, that's pretty massive undertaking. And what they did in this paper, right, was they were using genetic association, GWAS data, and other annotations and enrichments to basically discover new loci. And also discover for those loci are the factors in those loci that are in common that make them shared across multiple psychiatric traits. So it's another layer of evidence to help with target identification and selection of genes for functional follow-up. And they identified a bunch of new loci. So I looked at this and I was thinking about the work you're going to hear about today. And I was thinking about, wow, look at these beautiful figures. Look at all the data that's ingested in here and sort of encompassed in these figures, right? They have new loci. They did a new modeling of genetic overlaps with, factorization through genetics to present models of how these different traits are co-regulated and co-functional, and then do function analysis to look at like a functional enrichment on top of it all. So it basically provides not only loci for a specific psychiatric disease, but how they're related to others. So that's all this incredible information, right? They also provide analysis down to like the functional annotation, looking with those factors for the variants that they identified with the different psychiatric traits, adding in functional annotations so they can understand what cell types are, involved in those different disorders. So this is, as an expert, if you study psychiatric traits, this is a paper that you're going to really want to, understand and work with, because we know human genetics matter when it comes to, identifying targets, right? The traditional approach is you study something in a mouse and then you look in the human evidence. But really, the most successful treatments really come from using human genetics as the anchor, right? You discover genes or biomarkers or pathways with relevance to humans. Then you figure out the mechanism and then you make sure you use those studies to predict that efficacy. Right. So that it gives you more power. And that's a core tenet of the resources that we jointly build across the genetics knowledge portals, which you know about, and also GWAS catalog, right? They host GWAS data. That's the core tenet, right? So for common metabolic diseases knowledge portal and types diabetes beforehand, and more recently, the Association to Function Knowledge Portal, we work with communities to understand their traits, their data, to ingest that information and make that accessible to you so you can learn that. So like the data that you just saw me mention eventually will be integrated into the knowledge portals. However, the experts themselves really know it best, right? So our job is to enable it to be easier for you to take, GWAS data, run all these analyses, which you could do yourself if you know what you're doing, and then, help you weigh your evidence for a gene or a locus. That's really what we do with the portals based on, I have a gene of interest. I want to know what the genetic support is for it, or, tell me all the information related to my favorite phenotype, right? That's really what the portal enables. And that we do that by incorporating a host of research questions and proposals going from the genetic associations all the way to the functional, organismal output, but it requires a host of other resources and bioinformatic approaches in between that, right? So that's been in their job with what we built with these portals through representation. We make these data accessible through scientific questions and summary representation and visualizations. But our partner's GWAS catalog makes sure all that data is captured systematically in one place. And that's where you go for GWAS data, right? If you want to go for all the GWAS data for your favorite trait or phenotype, you go here and you can get all of it organized in one way. We think about summarizing that and integrating it. So with those two approaches, we get lucky, we get to work together. But the GWAS catalog has a pretty substantial mandate. They house all this data. And this is pretty crazy. But as a human geneticist or as a biologist, if you want to study this, there's hundreds and hundreds and sometimes thousands of loci for a given trait. It's pretty intense, right? And it's growing, right? You just saw another paper where we now have added more information for psychiatric traits, of how they relate to others. And that would be another layer of evidence, right? So, imagine if you want to know what genes you should study for your favorite area of research, or I study a gene, tell me what human genetics says about it. It's pretty complicated, right? Moreover, so we do this with our portals and we present to you sort of like a summarized score. We call this a huge score, where we take all the genetic data from the common and the rare and we layer it on and we make it accessible to you. But our portals are great, but actually you as experts are the best generators of this. So what we've noticed in our field, right, not only are we generating GWAS data sets, but in addition to GWAS data sets from different communities and consortia, we are also doing things to go one step further, which is, okay, great, we have a bunch of loci, but let's then try and say, for those loci, what are the predicted effector genes, or the genes that are causing the association, driving the association at a particular trait at a loci? So do that. Again, I mentioned all these layers of evidence, but folks who understand their trait, their cell type, their disease are really the experts to go to, right? So in many cases, you're getting these beautiful papers produced. Like this is an example of a very iterative and comprehensive approach, where this was a kidney GWAS done a couple of years ago. They updated with more samples. They also added enrichments, and they added essentially a scorecard for creatinine, EGFR. They basically were able to say for all their loci, basically prioritize them based on a set of evidence. And they made all that accessible on their lab website, right? Which is great. But it's not necessarily easily available at your hands, nor is it standard in a way you can either integrate into one of your methods, or like your pipeline analysis you do for your other work, or in a way that's systematic like the GWAS catalog does, where it's like in one place, standardized, easy to access. And our field really needs this, because folks who are biologists don't necessarily want to wade through all these different papers. They kind of want it brought to them. So we thought, given that communities are not just doing GWAS, and we are, as portal developers, are responsible for representing it, we also want to represent the curated results from all of these resources and research efforts. So for that, the field sort of turned to these gene prioritizations, where it's not only GWAS, right, but it's also evidence sources compiled into one paper and then shared. So our communities are doing this at scale as well. And so what we did is we started with an effort to basically say, let's go grab all of them that are out there. Let's do a GWAS literature search. Let's talk, GWAS literature search. Let's talk to our collaborators. Let's do a survey, and let's get everything we can get from collaborators. We obtained all this data. We sifted through this. I say we, but it really was done by Maria Costanzo initially. And we basically looked at, how do we get this all accessible to others? And in doing that, we're like, wow, it's crazy. There's so much different ways people capture things, different methods, different things people are doing. But let's get it in one place. So the first effort was just to get it onto a portal. We call this the PEGS portal. It's part of the CMDKP and the A2F portal. So it's all accessible in one place for these 175 gene lists. But the community works faster than we can, right? They're churning out these lists every day for the most part. We can keep the evidence sources in one place, but that's if we spent a lot of time, like hours, basically curating and formatting and gathering all that data. It's a lot of work. And we want to make it easier for collaborators who want to make that accessible. Moreover, if we want to make a resource, we have to have a way to standardize that. And we've gone, to some extent to be able to do this. Like if you're on our gene page now, if you scroll to the bottom for all these lists, it's integrated insofar as we say, like, the community experts have said that this gene is also involved in this particular disease for these reasons. And you can see the evidence sources. But what we observed in doing this is as the GWAS data goes up, so do the effector gene lists go up. But it's like the Wild West. Everybody has a way of formatting it, styling it, putting it in an auxiliary supplemental figure in their paper, not even making it accessible because they didn't think it was as important as the top findings, for example. But that list is very valuable because it has all of the layered evidence sources, and it can be used in many ways downstream for others. So to that end, we did this review where we looked through all the literature. We said, what are the different types of gene lists? How do they bifurcate in terms of like how people present them? What do they weigh? Are they bioinformatics? Are they heuristic? So on and so forth. So that was a review that we did, but this also prompted us to take an approach, which was to lean into the strengths of our two groups. One for us from the human genetics side in terms of representing knowledge and integrative results, and the other from the GWAS catalog side, thinking about FAIR principles, how to really ingest this data systematically in a format way that others can make use of. And so we sought to basically create a bunch of standards for this and work with you all as community members to raise awareness about the landscape, to establish a community standard, and really invite folks who are generating these lists to do those standards, but also participate in how to make them so that they would be easy for you to make them accessible along with your GWAS, for example, in all of your papers, or how to make it so when you have all these different supplemental figures, you can immediately point somebody to a resource where they can easily interactively query. So we really sought out to do this together. We sort of thought to make a straw man, to benchmark against things. We engaged stewards of a community, which you're going to hear about from Aoife and Laura. We are working towards a manuscript and a white paper summarizing these evidence sources. In fact, we're near close to being done on that. We also want to create a resource for the deposition and access of these lists for the ones we currently have and going forward to make it super easy for people to participate and contribute. And hopefully this will make a roadmap for us to go forward to make these more useful in other ways. So that's really what we did. It was an international team. It wasn't just an international team of folks I mentioned. It was really, this is the two teams across EBI and Broad. And I want to particularly call out before I turn it over to Aoife, who is actually going to present later, but this is also her last presentation with us. She is moving on to bigger and better things from her role in EBI, and it's been really a joy and a pleasure to work with her. She's really led some of this effort that you're going to hear about today, but not only that, she's also been a collaborator that has, really advanced our portals over the past couple of years. So we'll miss you, Aoife, and happy to hear from you later in this conversation. So the workshop that we produced in order to get folks involved had about 160 people, but moreover, it had like 7,000 views on YouTube, which means people care about it, which makes me super happy. We hosted this, made it all accessible. I'm just giving you a sort of summary of how we made this sort of like, not just about us telling the others what you should do, really about you as community members developing these resources, how you want them to be benchmarked and established as standards, what you think about is useful and important. So this all led to the framework you're about to hear from, which we lovingly call Pegasus. You'll hear what it stands for. But we're doing this with one other consideration in mind. So let's say we do this all well, right? Let's say that we make it super easy for folks to ingest their effector gene list whenever they have their paper. They send it off to the GWAS catalog. And in addition to their GWAS data, they also deposit these very simple formats of their effector gene lists. And that would then get integrated into the GWAS catalog. It would also get integrated into our portal. So you could search it along with your favorite gene, see it with all the other phenotypes we have, sort of an integrated place. But moreover, we're living in the world of LLMs and AI. And in order to make these lists useful, you have to make them standardized so they can be easily incorporated into machine learning models so people can learn from them and use them in their processes. I think this would be very valuable for us. So we're keeping that in mind as we're building this resource, that this resource will not only be a place where people just query, but people can actually extract information that we've learned about all these genes and all the evidence sources with them in a way that is structured such that an agent can actually synthesize information from it and make use of it and summarize it for you, the user. So we're trying to make context-based representations of all these results once we get them all together. That's the vision, and we hope to get there. But more importantly, right now, let's talk about the standard. I'll stop sharing.
Okay, so I'm going to take over, and I'm just to introduce myself. So I'm Laura Harris, and I'm the project lead of the GWAS catalog. And yeah, just to echo what Noelle says, it's been a real pleasure to work with these guys over the last year or two. And it's great when resources work together, right? Because you don't want to go to different resources and find we're all doing things in slightly different ways. It's definitely something that we want to contribute to the community and improve things by working together. So I am just going to run over in a little bit more detail the work that Noelle mentioned that was mainly carried out by Maria from the Broad Institute before she retired. Um, really outlining our motivation for why we wanted to develop this standard. Um, So yeah, as Noelle mentioned, so our motivation from the GWAS catalog. So at the moment we have this huge amount of GWAS data, but we only provide map genes, so the closest gene per variant. We don't include predicted effector genes because of this sort of lack of provenance, and they're not reported in a systematic way. And as Noelle mentioned, there was a huge effort to curate these in the knowledge portals, but that curation was very hard to do. It's very manual. There was a lot of missing data, and it's very difficult to make this data findable, accessible, interoperable, and reusable or fair. And so the goal for us is to develop a data standard, a fair data standard for PEG data that works both for ingest into resources and also more widely across the community, enabling things like meta-analysis to define gold standard lists of genes, integrating data and reusing data in computational pipelines and submission to knowledge bases, supporting the use of AI, and obviously the kind of the ultimate goal is to generate hypotheses and identify new drug targets. So I just want to talk a little bit about some terminology. So for the purposes of this discussion, we're defining a post-GWAS activity, right? So at the most basic level, you do your GWAS and you can say, here's my significant variant, and this is the closest gene. So this is the one I'm going to annotate to that variant. But the majority of researchers go a little bit further, and they perform an activity which we're going to call gene prioritization. So they take a bunch of evidence for different genes in the locus and assess that evidence to see which is the strongest. And commonly, but not always, they go a step further and produce a synthesis of all the evidence into a single display. So typically a table, they give a score to each gene. So it's really easy for the downstream user to say, yes, we predicted that for this variant, at this locus, we predict that this gene is the effector. And the first thing we encountered is that there's some confusion in the field about what terminology we use even to call these things, right? So there are a lot of different terms in use. We surveyed the community on which ones were most preferred, and we rapidly discarded the last three because they have some nuances of meaning that have particular meanings in different contexts. And the first two, causal gene and causative gene, are maybe a bit too deterministic. They have a certainty that may not be justified because essentially these are predictions. So that's why we've gone for predicted effector gene. We're using the acronym PEGs for these genes and the data as a PEG list. So the work that Maria did was to survey publications and try and find trends in the data and commonality between studies. So considering around 5,000 papers that were indexed in the GWAS catalogue over a 10-year period, we looked for papers that mentioned gene prioritization, either in the title or the abstract. and then reviewed those to just check that they did in fact meet our definition of effector gene prediction. So aggregating multiple evidence types and doing that systematically across all significant loci from a GWAS rather than specific candidate ones. And at the end of that, we had 169 papers covering 157 traits. And as Noel already mentioned, the first thing we noticed is that the percentage of GWAS papers that include gene prioritization has really increased year on year, and it's only continued to increase since then. So we can divide the types of evidence into two kind of very high level categories. So firstly, variant-centric evidence. So this is where something like statistical fine mapping has been performed on the GWAS data to predict variants which are most likely to be causal, and then considering those causal variants, finding evidence about their impact on gene function. So, for example, considering the known effect via the VEP, whether it's in contact with regulatory regions, whether it's a QTL, and using that to build up evidence for gene function. The other way to do this is to take a wider definition of the GWAS loci and look at all the genes in the loci and find evidence about their function and do it as kind of a top-down process. So looking at membership of a gene set, perturbation evidence, gene burden analyses, and general kind of knowledge from the literature about that gene function and find out which ones are most relevant to the phenotype. Then, of course, there are many bioinformatics pipelines that are used either in isolation or they might be combined with independent evidence types and used to build up evidence in that way. So first question was to see whether there was any kind of pattern or commonality between studies. So could we say, a certain subset of studies had this particular mixture of evidence to them? And we rapidly found that there were just many different variations. So given these kind of 8 high-level evidence types, some papers included all 8 of them, some included only 1. The most common number was 4, but there was no real pattern as to which ones were included. Of the 169 papers, there were 73 distinct sets of evidence categories, and the most commonly used ones, so the variant location and QTLs, they only co-occurred in 10 of the papers out of the 169. So we couldn't identify any clear trends in the use of different evidence categories. We couldn't see any trends over time. We couldn't group studies into distinctive types. So then looking at the data presentation, so we considered whether authors only did gene prioritization, so they just presented different evidence types in different tables, or did they go that step further and integrate all the evidence into a PEG list? And we found that 25% of papers only included the gene prioritization and 75% went that step further and actually predicted effector genes. However, of those, around 10% only presented the data as an image. So and in many cases, these were just graphics presented without their underlying data. So this obviously makes it very difficult to kind of reuse and present in that for ingestion into a resource, as we're kind of aiming for. In some cases, evidence was presented for all genes in the locus. So, for example, here there's almost 20 genes in the locus that were under consideration, but in around 30% of papers, only the top gene for each locus was presented. So it wasn't possible to tell what the full range of genes in the locus considered was. Around 30% of papers gave a scoring system, so it was very easy to see whether the predicted gene in one locus was equivalent in strength to a predicted gene in another locus. But in 70% of papers, there was no scoring system, so that the data was just presented as it is. There was also a lot of variation in how the locus was defined, which is quite a sort of straightforward thing, but this complicates comparison between data sets. And even when the locus was defined as all genes within the certain distance of the sentinel variant, there was a huge variation in the range of distance that was considered. Some authors combined LD and distance measurements. Some annotated with the GWAS index variant, some didn't. And when they were annotated, it was different methods were used to identify the variant. So the conclusion from this work is really that there is substantial heterogeneity. There is no kind of most popular way to do this analysis, and there's no most common way to represent the data. Of those 169 papers that we analysed, only 115 of them presented data in a tabular format that was potentially reusable. And then of those sort of basic requirements, like whether the loci were identified, whether all the genes were presented, and whether scores were assigned to the genes, were not present in the majority. So only 19 of them actually included all of those things. So what does this mean in practice? So really, one of the things we want to do with this data is we want to be able to compare predictions for the same trait. So we want to know, do two different analyses produce the same effector genes, and can we consider those gold standard genes for the trait? So of those 169 papers, there were 49 where there were multiple analyses of the same trait. But 41 of those didn't provide either a definition of the locus or that they didn't provide a scoring metric. So it wasn't possible to do that comparison. And in the end, we only had 4 pairs of papers that could be compared. So for the two papers for heart failure, as examples, we had one paper which had a table showing the loci. There were multiple genes per row, but with no score. So that needed some manual table wrangling to get to 1 row per gene, and then another manual step to add a column with the sum of the number of evidence types. So all the information was there, but it just needed a bit of downstream work to get to a point where we could make the comparison. And then the second list we were comparing had a graphic showing evidence for each gene with the top genes listed there. It didn't define the loci, but that was done in a separate table in the paper. So again, that needed a little bit of wrangling to get the data together. And then in the end, we were able to find out that of 9 loci that were shared, so this was 2 different GWAS, so they identified 9 of the same loci. And of those 9 loci, the same gene was identified at 6 of them, which is a really important result. And that's really valuable data for the community. But it did take a lot of effort to get to that point where we could make that conclusion. So as Noelle already mentioned, we published the paper in Nature Genetics last year outlining all of this work, and I really encourage you to read that if you're interested in it. And based on this analysis, we made some high-level recommendations for reporting effector genes. So really things like the data should be in a single file in a tabular format. It doesn't mean that you can't also make a lovely graphic, but all the underlying data there should also always be reported. And there needs to be consistency in how the genes are defined, the identifiers that are used, and so on. But we also recognize that these are very high-level recommendations, and we really need to focus in detail to come up with an actual usable template and data standard for researchers to use and apply in their publications. So we had our initial community workshop in September 2024, as Noelle mentioned, and the recordings of that are still available. So that was organised by the Knowledge Portals team and by the GWAS catalogue team. We also had open targets there. So, we have a lot of interest across resources. Based on the discussions at that workshop, we produced a draft standard, and we then set up a working group, a more focused working group of around 15 to 20 people, and we met 5 times over the course of the year to iterate over the standard, to focus in on the different parts of it, and to perform a benchmarking activity. So we asked the working group members to reformat either their own data or other people's data into the proposed standard, and get feedback on whether it was useful and workable, and how we could improve it. And then, of course, we launched it at ASHG in October. So I'm going to stop there and hand over to Aoife, who's going to talk about the details of the standard. If I stop my screen share.
Not mine. Okay. How does that look?
Looks good.
Okay. Right, thank you, Laura. Yeah, so I'm going to present basically the outcome of all of this work that we've been doing over the last couple of years. And yes, as Noelle alluded to, we have decided to call this framework Pegasus. So predicted effector gene, aggregation standards, and unified schema. So as Noelle and Laura mentioned, this is a collaborative effort between ourselves at the EBI, where we produce the GWAS catalogue, and our lovely colleagues at the Knowledge Portal network at the Broad. But I also want to acknowledge the contribution of the members of the working group who've been at the working group meetings, the attendees at the workshop, including our colleagues at Open Targets. So, as has been mentioned, our goals were our main aim was to develop a framework, a standard to make these predicted effector gene data more findable, accessible, interoperable, and reusable. So as Laura pretty thoroughly covered, currently they are not any of these things. And by making these data more fair, we hope to build a foundation for our ultimate long-term aim, which would be to enable meta-analyses of this data to define gold standards of lists of genes that are involved in these traits. So basically, what is the underlying biological mechanism that's driving these genetic associations? So yeah, we were considering these specific use cases that data integration and reuse, they should enable computational ingest, submission to our knowledge bases, and support the AI use case. And by exposing the biological, basis that they support research and hypothesis development. So these are the goals we're working towards. Before I get into what we came up with, I want you to put you a little bit in mind of like our perspective as we were going through this process and thinking about how to develop this standard. So it would be wonderful if we would be able to come up with a standard or a framework that could achieve all of these three corners of this triangle, so that we produce something that achieves extremely high data quality, and by quality we mean fairness. We could do that while maintaining a low burden on the data generators. It would be pretty easy for them to generate, and that we would be able to do this with a low curation cost. But it's kind of one of these pick two of three scenarios. Each two that you pick has implications for the third. So as we were going through developing the standard, this is what we were thinking about. So if we say curation was covered there to get the lists that are in the portal right now, a lot of curation was required. But what if we wanted low curation cost? What we currently have is high curation cost. There's a low burden on the data generator, and to make the data more fair, it requires a lot of curation. And even that curation currently doesn't make it fair enough. But so if we go down this route, we need to consider, will curation be possible going forward? As Noelle mentioned, curation is super expensive, and you need to consider resourcing in our current constrained funding environment. So if we flip that, we take the burden off the curation, that means we need to put more burden on our data generators. And when we do that, we need to consider how the data generators are being incentivized. Why will they go to the trouble, the effort to put their data into this format? And then you need to think about, do we need to get journals involved to be able to mandate that they do this? Are there other incentivization methods? Will people be incentivized by people using their data more? All of those kind of things. The other scenario is that we compromise the data quality somewhat, reduce the burden on the data generator, and reduce the curation cost. If we do that, we need to consider how good is good enough, how fair is good enough. And thinking about that, we need to think specifically about what our use cases are and what our users really require. But really, it's not a choose two scenario. What we've been trying to achieve is a balance between all of these different factors. So just try and consider those things as I'm presenting what I'm showing you. So the Pegasus framework we've put on GitHub. Can somebody put this link in the chat so that you can follow along if you wish. So the link here will bring you to a specific page in our documentation, which is our toy data example, but there's a lot more in there. There's a whole background and overview of the framework, more detail on all the different components, as well as lots of background material. The community at the working groups are all recorded, the meetings were all recorded. There's posters, there's all sorts of stuff in there to explore. And our slides, our materials from ASHG are in there too. So yes, this is all on GitHub and publicly available. So to illustrate the framework, I'm going to be using this toy data example. So this is just a little piece of synthetic made up data that's kind of representative of the kind of data that we see in publications. We've just got a pretend trait of myocardial infarction. We're looking at two different loci that have been identified by two GWAS significant variants, variant 1 and variant 2. The authors in this pretend paper have done a range of different analyses, different evidence streams of the different genes and variants in the region. But as is typical, this data would be distributed across the paper in different places, potentially in supplemental tables, text, figures, all of those things. So our authors have done a couple of evidence streams, EQTL, LD. They've looked at knockout models to see are there any relevant phenotypes and mouse models, so like a heart phenotype, for example. They've looked at expression levels in a relevant tissue. They've done one of the bioinformatic methods that Laura mentioned. POPs is a popular method for integrating different evidence streams. And then they also have their own conclusion that they provide. So this is kind of our like toy example for thinking about how the framework would be applied. So the core of the framework, it's a tripartite framework. It's basically got 3 modules. This is the absolute core of the framework. So we have a metadata file, and metadata is data that describes data. Then you have an evidence matrix, which is a potentially large file that's got lots of all the evidence in it. And then a summarization of that extensive resource of data into a high-level predicted effector gene or Pegasus list. So PEG lists must be supported by an evidence matrix, and that must be described by metadata file. So the idea of the distinction between an evidence matrix and list didn't really exist in the literature or in like the landscape analysis that we performed. This is something that really came out of our initial workshop in September 2024. And yeah, so in the metadata file, what you will get is descriptions of the trait and the provenance of the original underlying GWAS data. Remember, this is a post-GWAS analysis. It will specify things like the sources and the provenance of all the different evidence types used. It should include a description of the methods used, and importantly, have definitions of the column headers for all of the other data files. The evidence matrix should include evidence for all of the genes considered, not just the top genes. As Laura mentioned, that there's a lot of variability in the landscape currently, but for this framework, we're requiring evidence for all of the genes considered, and it should include data for all of the evidence types used. It may include integrations. So integrations are authors integrating different evidence streams together. So an evidence matrix could exist without an integration, but if you want it to be represented as a list, it must include an integration, because the integration is what the summarization is based on. So the Pegasus list is a high-level data summary. So this is basically like, what's the answer at the end of all of those workings? It lists the best predictions for each locus, so the top gene at each locus. That's what is included in the list. And it indicates which evidence categories were assessed in the analysis. And crucially, it includes an author's conclusion, which is an integration of the evidence, and that defines what makes it onto the list. I'll go into each of them in more detail. But if you take only one thing away from today, is that the Pegasus framework is this tripartite framework that's got 3 modules: metadata, evidence matrix, and list. Just a little bit more details for it to be represented in this framework. It will be a representation of 1 trait, 1 GWAS, and 1 matrix. And the data in each of the different modules is organized into kind of 3 sections. The first one is quite descriptive, describing the variant, the locus, and the gene. The next section is about the different evidence that's included, the different evidence streams, and they are assigned to categories. And the categories are specified as variant-centric or gene-centric, as Laura described what that means. And then we have a section on the results of integrating the different evidence streams. This organization applies across all of the different modules of the framework. So to consider our toy data, this is kind of how that would be divided up, organized. The first section would have description of the GWAS, the trait, the variant, the gene, and the locus. And then this section are the different separate evidence streams divided into variant-centric and gene-centric. So in this case, EQTL and LD apply to the variant. The perturbation, the knockout model of the gene applies to the gene, and the expression applies to the gene. And in this example, the authors have done 2 integrations, the POPs, and then they have their own author conclusion. So the evidence categories that we've defined were based off the landscape analysis that we performed. So these are the things that people are doing. They're a refinement of that slide Laura showed you earlier about the variant-centric versus gene-centric evidence streams. There's more detail on these in the documentation, but you can see the different kind of things that people are doing, and I think they've been covered already. One thing that we've been working on post our presentation at ASHG is mapping these categories to ontology terms. So the mapping of the different evidence streams to categories really helps to integrating across different lists. So you could imagine how different specific evidence and analyses types could be categorized, and then you could integrate more easily together. But mapping to ontology terms really strengthens your ability to be interoperable with other resources and to future-proof as new things are added. So then the metadata, we've defined a metadata schema, again, following the same 3 sections. The trait is described ideally by an ontology ID. The GWAS data, you can specify using the GWAS catalog accession, GCST. If not, yeah, you need to describe it in a little bit more detail. And then metadata on the variant, locus, and gene is around, what's the genome build? How did you define the boundaries of your locus? The evidence streams description in the metadata can be quite extensive. As I mentioned, you'd say, is it gene or variant centric, and it's how it's categorized. There will be description of the provenance, where does it come from, what method was used to analyze it, what's the biosample, what tissue does it come from, and the mode, is it human curated, is it computational, or is it a mix of the two? And then the integration is how and which evidence streams were integrated. Sometimes people do lots of different integrations, trying to integrate different combinations of evidence. The method by which the evidence streams were integrated, and again, was that human curated? Was that computational? If so, what's the software? All that kind of stuff. And crucially, for everything, definitions of the column headers and which, for example, evidence streams they apply. This is our schema. This could be represented either as a YAML or a JSON, so a machine readable file. Just to give it a little bit more detail, this is kind of a zoom in on the kind of a more formal representation of the evidence schema. So you can see the kind of things that would be included. So for the source, there'll be a range of specified fields describing where it came from, the version, the URL, all that kind of stuff. And again, column header definitions and to which evidence stream they apply. This is an example of what the metadata would look like for our toy data example. So for example, this is for the eQTL data. That's in the molecular QTL category. It's in variant-centric data, and then you would describe the source and have additional information. This, for example, comes from GTEx, specific version, the method, it's fastQTL, have a version number, URL, DOI if applicable, and then there's 2 columns that apply to this evidence stream. If you go to the documentation on GitHub, you can see the full metadata example in YAML. As I said, we can represent it as YAML or JSON. We're using YAML in these examples because it's more easily readable for humans. So that is that. What is not easy for humans is generating a YAML or a JSON file. And this is probably the bit that's most involved from a data generator perspective. So what we've done is we've created a metadata data entry template. This is a spreadsheet-based data entry template built off the kind of modular nature of the metadata schema that should be, fairly straightforward to fill out. And this is based off our experience at the GWAS and PGS catalogs in providing these kind of templates for people to do data ingest. So that's the metadata. The next one, the evidence matrix. So again, this can be the potentially large file where all the evidence lives. This is just to give you an overview of how this is organized. Pretty basic, the variant ID, chromosome base pair ref alt, and then the locus information, so the range of the locus that is being considered. Again, that was something that was often missing and really hampered meta-analysis. And then gene information, sections on all the different evidence streams. We're suggesting that the header should include an abbreviation of the category to aid with human readability and creating the files. And then the section where all your integrations would be. Now, as I said, integrations are technically optional if you can just have an evidence matrix without any integration, but they are absolutely mandatory for representing the data as a list. And again, here's our toy data in the format. Like I said, pretty straightforward. But you can see that the clear advantage of this is that everything is in the same page, in the same place, and you don't have to hunt across multiple supplementary tables to pull it all together. So the next thing then is the Pegasus list. Again, this is a high-level data summary. So this is for the person who just wants to come and see what's the answer, what have people found as being the top genes at GWAS loci for this trait. And this is the Pegasus list for our toy data example. So again, we had 2 loci. So on our list, we've got top gene for each locus, so that's 2 genes. So it's pretty simple. We've got the variant, that was the lead variant, the gene, and then just an indication of which evidence streams, or which evidence categories, were included in the analysis. This doesn't speak to whether the evidence was positive or negative. It just gives it a fairly lightweight overview of what types of evidence were considered in coming to the author conclusion. So that's the other thing that needs to be on the list. The author conclusion is one integration from the evidence matrix that is specified. The authors must specify it as their conclusion. And then that's the one that will be the top one in that integration will determine what makes it onto the list. So for our toy example, we've got strong for gene 1 and moderate for gene 4. You can see that, so we were calling this our version 1 model, and you can see how future iterations could expand on this with much more detailed annotations, more structured scoring, potentially a way for authors to specify a threshold for what makes it onto the list or not. But for now, we're going with the most straightforward implementation, which is this version 1 model, so top gene per locus. So that is the framework. So to summarize, we're proposing this Pegasus framework, which is composed of 3 modules. We hope that the 3 modules capture the richness of the underlying data while communicating the high-level findings very straightforwardly to people who just want to access that. We feel with the standardization and the categorization that this enhances the interpretability and enables more interoperability of PEG data. We hope that it supports different use cases. So we've got the underlying data, so that's good for data reanalysis and meta-analysis, but we're also supporting human readability and machine ingest. We feel it facilitates integration of the data into the knowledge bases, the PEG portal that Noel mentioned, and linkage in the GWAS catalog. And you can see how it lays the foundation for meta-analysis and a universal scoring system and the generation of gold standard effector gene lists. So even on a very simple basis, you could see if you had multiple PEG lists for a single trait, you could very easily generate a gold standard list for that trait. And that is all I have for today. I think we have a couple of minutes for questions. Let me stop.
There's one question in the Q&A that basically is saying, is Pegasus capable of distinguishing the effects of metadata variables such as sex or age on a given trait? For example, can Pegasus indicate whether a predicted gene trait association score is consistent across sexes or across different stages of the lifespan?
I don't think we've included sex as a specific metadata item, but it could easily be included. I think lifespan, again, it would depend on the analysis and what's been done, but I think the high-level answer to that is we would hope that the structuring of the data would help people address those questions broadly, and questions like that in a way that they couldn't without the standard. But yeah, I don't think we've included sex specifically as a metadata item. That's a good point.
So if I can just chip in, Aoife, can you hear me okay?
Yeah.
Yeah, so sex is included in the GWAS catalog summary statistics metadata format. So if the GWAS data is already has all the required metadata there, so yeah, then you would be able to do that.
Thanks, Laura.
Any other questions? Please feel free to use the question and answer, the chat, or raise your hand.
There's a question about where to find the recording of this webinar later.
Yes, so I responded individually, but I will put this in the main chat. Basically, we have a YouTube page, and I will also send out the link from the YouTube in an email and share over our social media as well.
And I think we'll also add it to the GitHub, the documentation that we've got on the GitHub. We have a community page there where you can find all of the working group meetings, all of the materials from the workshop, all that stuff. So we can put it there as well.
Perfect.
All right, if there's no other questions, we can just quickly wrap things up. So thank you so much to all of our panelists for this incredibly informative session, to the amazing working group that contributed to this effort over the course of the last several months. As Aoife and Laura and Noel said, this has been a large international collaboration with input from experts from across the field. So we're really excited to be launching this. Stay tuned for some of our next steps. We're going to be putting out a white paper or manuscript describing some of the outputs of this working group.
we're putting together a resource that enables deposit of these lists so that you can access them in a centralized place once they've been formatted according to the standard. And eventually we'll make it so that the list can also be automatically contributed to well-known repositories like the knowledge portals or the GWAS catalog. So stay tuned for that. And then finally, we're developing a resource for the presentation, curation, and interpretation of these lists. So that's going to be a longer term sort of effort, but we hope to develop out some additional tools to enable that as well. So if you'd like to submit your own peg list and or to collaborate on further efforts, we are looking for volunteers from the community. So we would love to hear from you. This is our email address right here, help@kp4cd.org. So if you have a list that you would like to try out the standard on, let us know what you think. Feel free to reach out and we would be happy to work with you. So with that, thank you all for your participation, and we look forward to sharing with you the next steps and the white paper once we have it available. So, thank you.
And thank you to our speakers. We greatly appreciate it. Hope everyone has a great end of the year and happy holidays. Thanks, everyone.
Thanks, Mackenzie.
Thank
Bye,
you.
everyone.
Bye.
Bye-bye.